Thank you. Hello, everyone. Today, I will be talking about reclassifying variants of uncertain significance with transcriptional profiling. I'm a postdoc at Dr. Hannah Carter's lab, one of the moderators today at UCSD, and I'm excited to talk about some VVSs, but I'm going to provide a brief background, not too much, as it has been extensively described before by the great talks before me already. So variants can be classified according to their clinical significance as pathogenic if they show clinical effects, or benign with no effects. However, information about consequences of many are lacking, leading to a variant of uncertain significance, or VUS designation, which presents a challenge for genetic tests and their clinical interpretation. And these VVSs constitute the largest portion of human genomic variations, making up more than 1 million variants in the ClinVar database. So resolution of these VVSs is critical for precision genomic medicine. And RUNX1 gene is one such case where half of the variants are of uncertain significance. And familial platelet disorder, FPD, also known as RUNX1 FPD or FPD AML, is a rare inherited disease caused by a germline mutation in the RUNX1 gene, which has a 50% chance of being inherited by the children of an affected parent. And it results in lower blood platelet counts and platelet dysfunction observed more than 90% of the patients, which can cause easy bruising and prolonged bleeding after injuries. And the patients also often face a range of health issues like asthma, allergies, autoimmune disorders, and gastrointestinal problems. But more importantly, 40% of the patients are at risk of developing hematological malignancies, blood cancers, throughout their lifetimes, like MDS or AML, through clonal expansion of acquired somatic mutations. So reclassifying these VVSs is a major challenge in precision genomic medicine, and there exist many state-of-the-art computational tools to identify variant effects. Many of these tools focus on the protein level effects of variants, but it's being increasingly evident that different mutations targeting the same gene can have different effects on protein activity and target different cellular programs, which raises the question, can we study same gene mutations as perturbations at the systems level as opposed to the protein level, based on how they impact the underlying molecular interaction network architecture and hence the cellular programs and states? And here we focus on RUNX1, the main driver of familial platelet disorder, as I mentioned, which is part of a transcription factor complex consisting of the DNA binding RUNX1 and non-DNA binding CBFP proteins. And RUNX1 is a master regulator of hematopoiesis, and as such, it regulates cell proliferation and differentiation. So it's the key regulator of cellular identity and function. And many of its functions happen through its runt domain, where it binds to promoters and enhancers, binds to CBFP and other transcription factors and co-regulators. And the importance of this domain is reflected by how frequently it's been mutated in FPD and leukemias, especially AML. So here we wanted to understand the potential of different mutations on RUNX1 to generate distinct functional effects in terms of how they modify cellular programs and states. And Perturb-6 provides such tool to investigate perturbation effects, ranging from whole gene knockout to single amino acid substitutions. And we use SEUS, a Perturb-6 approach developed by Dr. Pashan Mali's lab at UCSD, which uses bar-coded open reading frame overexpression libraries coupled with single-cell RNA sequencing to assay cell state and fitness. And the key point here is that each library element contains a unique barcode so that each cell can be mapped to a single element after sequencing. So we started by creating a structure resolved interface map of the RUNX1 runt domain responsible for many of its interactions, where each row represents the amino acid sequence of the runt domain, and the interface residues that are involved in physical interaction with each protein partner are highlighted in black. And we were able to structurally resolve 33 interactions of RUNX1 that had available 3D structural data. So here we can see the RUNX1 subnetwork we created based on this map. And within the runt domain, we identified 83 residues that perturb at least one interaction of RUNX1. So we designed an open reading frame library to perturb each residue. The single-cell experiment was designed to overexpress a vector containing the wrong-sum variant, followed by a barcode unique to each variant after force identification after sequencing. And we use a chronic myeloid leukemia cell line where RUNX1 is a regulator of cell identity and contains wild-type RUNX1. And we repress endogenous RUNX1 to see the mutational effects more clearly. So we created a clonal cell line with doxycycline-inducible CRISPR knockdown of endogenous RUNX1, where we achieved about 70% repression in expression at both RNA and protein levels. The library contains 117 elements, including the wild-type and loss-of-function constructs as controls, the 83 perturbation variants, some combinations, and additional negative and positive control elements. So all these elements were sequenced in the single-cell setting, and we hypothesized that variants with different effects on RUNX1 targeting should cluster separately in the transcriptomic space, and we have the wild-type and loss-of-function controls to provide a frame of reference for functional impact. After quality control filtering, we recovered around 20,000 high-quality single cells with detectable barcodes mapping to a single variant only. And to better quantify the variants, the differences between variants, we pseudobulked them and compared their mean gene expression profiles. So while on top you map each dot represents a single cell, on the bottom each dot represents a single variant. And unsupervised clustering of variants revealed three groups that separated wild-type and negative controls from the loss-of-function and positive controls. And the separate assignment of some of the perturbation variants in these middle clusters suggested a putative hypomorphic effect, distinct from a complete LOF or wild-type activity. So we label these the variants in these clusters accordingly, and revisiting the single-cell visualization with these labels shows that we can see differences of functional phenotypes in the single-cell setting as well. To quantify transcriptional effects of mutations, we used Hotelink's two-sample t-squared test, which is a student's t-test generalized for multivariate testing. And we compared expression profile of each variant against the wild type or loss of function control, and used the T2 test statistic as a distance metric where higher scores indicate more deviation from the control. So this test revealed that all wild type-like variants are indistinguishable from the wild type control itself, with very little T2 scores against the wild type, but high T2 scores against the LOF. And the opposite is true for all LOF-like variants. However, hypomorphic variants are significantly different from both controls, suggesting that they result in transcriptional changes distinct from both of them, which is supported by differential expression analysis as well. Next, we evaluated cell fitness and saw that LOF-like variants occupy positive regions, while wild-type-like variants occupy negative regions with hypomorphic in between. So it seems like functional variants, which are LOF-like or hypomorphic in this case, are associated with increased fitness and proliferation, but also with upregulation of heme biosynthesis pathways, which is consistent with a shift towards erythroid differentiation with a loss of RUNX1 activity, in line with RUNX1's role in familial platelet disorder and leukemias. So next, we evaluated RUNX1 mutational landscape in cancer as a sanity check using mutations from the COSMIC and MLL databases. MLL is a strictly AML-based cohort. And we saw that functional mutations are significantly enriched in cancers, especially in MLL, which is consistent with RUNX1's significance in AML, but also depleted in NOMAD, which is a reference population, which is great. But next, which is our main aim here, is we wanted to see if our transcriptional profiling can help resolve the ambiguity around variants of uncertain significance. So we collected germline variants from the ClinVar database, and 24 unique variants overlap with our library. where we saw that our transcriptomic profiling correctly classified the pathogenicity of 8 labeled variants. So 1 benign and 7 pathogenic. And one of them is identified as hypomorphic and the rest as LF-like in our transcriptomic assay, giving confidence to our identification of the remaining 16 variants of uncertain significance. So here we identified 9 of them as wild-type-like, 3 as hypomorphic, and 4 as LOF-like. And you can see that our T2 score really very nicely separates the pathogenicity of these Kaplan-Meier germline variants, which is great to see. So far, we have analyzed over 100 RUNX1 rand domain variants, which is a good number, but there are over 2,000 possible substitutions on the RUNX1 protein. So we wanted to see if we can extend our analysis of the rand domain variants by predicting the functional impact of the remaining possible variants on the rand domain using our transcriptomic readouts from our library. So we have a data set of 79 perturbation variants, which we divided into a negative and positive class based on our transcriptomic-based phenotypic labels. And we trained a random forest classifier using 85 amino acid-based features from the Cinebox database, which is traditionally used for variant classification tasks. And we obtained a reasonable area under the ROC and precision recall curve scores around almost 0.9 on our test set, which was higher than using just sequence-based variant effect prediction from West or protein stability prediction from FoldX, which is great to see. So, but we have to also take into account that this is a very small data set, so it's very difficult to do cross-validation or anything. So, you know, we have to take that into account. But we proceeded to train on the entire data set and predicted the functional effects of all remaining rand domain variants. And again, first, we, as a sanity check, we looked at it in the cancer case and saw that our functional predictions are highly enriched in cancer, especially in MLL, but depleted in NOMAD, which is perfect. But then we wanted to assess performance on a high confidence data set. So here is a data set of pathogenic and neutral rand domain variants that are not in our training set, and we collected them from Cosmic MLL, Kaplan-Meier, and NOMAD databases. And this data set provided a reasonable area under the ROC and precision recall curve scores, which is great. And finally, we analyzed the remaining germline variants from the Kaplan-Meier data set. So, and we saw that we receive around reasonable accuracy on the 21 labeled variants, which gives confidence to our prediction of the remaining 103 variants of uncertain significance. Here, it is important to note that we are using a conservative threshold score of 0.5 to separate the pathogenic from the benign variants. But as you can see, actually relaxing the threshold towards the 0.4 range could increase accuracy because we have 4 false negatives and 3 of them will be correctly identified if we change the threshold. But so these are need to be taken into account, but we proceeded with 0.5. So yeah. In conclusion, in our study, we designed an interface-guided perturb-seq library and measured the impact of over 100 rand domain variants on more than 40,000 single-cell RNA profiles. And we showed that single-cell transcriptional profiling can identify functionally distinct groups. So in our case, it was 3 groups, wild-type-like, LOF-like, and hypomorphic variants, but a different gene can provide a higher range of phenotypes reachable by single amino acid substitutions potentially. And we show that this transcriptional profiling can inform reclassification of VUSs. In our case, it was 16 variants of uncertain significance, but we trained a classifier to predict effects of 103 more VUSs. So overall, this study demonstrates the potential of targeting protein interactions in the transcriptomic space to better define the landscape of cellular phenotypes reachable by missense mutations. And I want to conclude by thanking my advisor, Dr. Hannah Carter, and our collaborators. And here you can actually, our paper was recently published in Cell Reports, so you can like take a look at it by this QR code if you'd want. And I'm happy to take any questions. Thank you.
Done? Yes. Yeah, thanks. That was a great talk. The MLL data comes also along with transcriptome. I don't know if you had the opportunity to look at whether the transcriptome in the patients resemble what you see in your single cell assay.
I don't think we received the transcriptome data for MLL. Do you mean, was it bulk RNA-seq? I think, right?
You have bulk RNA-seq of the, yeah.
Okay, yeah, I will be interested in comparing the results, but I don't think I I obtained it while I was writing the paper. But thank you so much.